called statistical quality control, and our aim is to analyze the quality of the products that is manufactured in a manufacturing plant. So in a manufacturing plant, before the products are supplied to the market, we will do the quality analysis of these products. Suppose there are 500 nails manufactured in a plant. So we cannot analyze all these nails. You cannot do quality control of all these nails. So what we will do is that we will take few samples of these nails and we will do quality analysis of these samples. So this method is called sampling. So in sampling, you can see that here there are 50 nails. These are one sample consists of 5 nails and there are 10 samples. So we are going to do quality analysis for this 50 and we are going to plot a graph. So from this graph we can say that the entire batch is acceptable or not based on the quality analysis of these samples. So now what we are going to do is that while we are plotting charts, we have to plot two charts. They are X bar chart and R chart. So X bar chart means X bar is the mean of the thickness of these nails. Suppose we are taking a nail. So the thickness on approximately the center part of the nail is considered for analysis. So what we will do is that we will measure the thickness of these nails using a laser micrometer. So these 5 samples will be measured. The thickness of these 5 nails will be measured and we will mark it in the table. And we need to find the X bar, that means average value of thickness of one sample. So that is called X bar. So like that, hence X bar should be calculated and we have to plot these 10 X bars in the graph. So the X axis will be considering number of samples and Y axis will be having X bar units as values. So after plotting the X bar graph, we have to calculate the upper control line and lower control line for these nails. Upper control line means that the upper limit of the thickness. That means any value above the upper control line should be rejected and any value below the lower control line should be rejected. So that is the quality analysis, quality control. So after plotting X bar graph, we will calculate upper control line and lower control line using the equations given in your manual. And similarly, we have to plot R bar chart. R chart. R chart means R is the difference between highest value and lowest value of thickness. So 10 such R values will be obtained from this table, and also we have to calculate the R bar for substituting in the equation. So the equation is given in your manual. From that we will find out the upper control line and lower control line of R chart and we will plot it in the graph. So let us see how the experiment is done. First we will take a needle, and this is called laser micrometer, and we will place this in this 3-point chart. So we can see that a laser light is passing through this needle. So the laser light should be approximately in the center part of the needle because the needle is in a tapered shape. The thickness will be somewhat higher towards the head and somewhat lower towards the bottom. So that's why we are keeping approximately in the center part. So we are measuring the thickness of body in the center part, and you can see that the value is changing according to the thickness. So 1.88 is our obtained value. You can see that there are 4 decimal places. That means the least count of this instrument is 0.1 micrometer. So we have to mark all the 4 decimal points in your table also. So like similarly, you have to measure all 50 thickness of all 50 needles and then plot it in your graph. So this is your experiment.